Okay, what we did on Monday then was to look at the structure of blood, referring to the handout that you've got with two tables on it, but also the PowerPoint. So we went through, we had an overview of plasma, which of course is mostly water. But then of course you've got all the materials which are suspended or dissolved in that water. We've got a variety of proteins, and some of these we're going to be coming back to. So the antibodies in that list particularly, those are one thing that we're going to look at this morning. There was also a wide variety of ions floating around in the plasma for various functions, and we know what some of those are for. The sodium, potassium, calcium, for example, involved in nerve impulses and synapse function and all that sort of thing. And for the hydrogen carbonate there, the negative ion, we're going to come back to that as a start this morning, because I'm going to talk about how carbon dioxide is carried in the blood. And then there was a variety of other stuff floating around in there, depending on levels of nutrients, hormone requirements, that sort of thing, waste products. You'd find those there as well. So we then looked at the structure in terms of cells, and we have three different types of cells. We had the red cells, the erythrocytes, with their hemoglobin, very, very small, and with this biconcave shape, and important really to relate that to Fick's law, like we did with the alveoli in the lungs. You know, it's about having a large surface area and a small distance for diffusion, but also the business about the concentration gradient, maximising that. I forgot to add with this really that the hemoglobin actually reduces the oxygen concentration inside the cell so that more can diffuse in. If you think about it, because oxygen as it goes into the red blood cell isn't then just there in solution, so you haven't got a concentration of oxygen in the red blood cell because it's actually chemically bound to the hemoglobin. It becomes part of something else. So you've actually got effectively a low concentration of oxygen inside red blood cells to enable the oxygen to diffuse in. So all the three parts of Fick's law you can apply to red blood cells. And there is a question in the exam which basically says, describe how red blood cells are adapted to their function. And it's all these sort of things. It's the size, it's the shape, it's the hemoglobin. All right. And you've got all this on the sheets, but this was just sort of pulling it out in a different sort of format. Actually, I'm not sure that, no, you hadn't got all of the stuff about red cells on there. The white cells come in a variety of different types, and we will have a look at them down microscopes at some point next week probably. The ones that we're going to come across this morning as we go through the immune system are particularly the lymphocytes. The neutrophils are involved in engulfing and destroying foreign particles, microorganisms, that sort of thing. But they're not something that we're particularly going to focus on. But lymphocytes and their production of the antibodies are mentioned in the plasma. Those are going to be important. The white blood cells are bigger than the red blood cells, quite significantly so, but they do vary in size. Different ones are different sizes. They don't need to be as small. They are much squashier, and they can actually squeeze out of blood vessels and travel around in the tissues, the neutrophils particularly. You get white blood cells actually sort of migrating into the tissues to attack infection in tissues, and you get, if you get an infected, say, hair follicle or something like that that goes pussy, pus is actually a mixture of white blood cells, bacteria, and cell debris, and it's because the white cells have actually managed to get out. The white cells then, got a variety of roles. The thrombocytes are platelets, and those are involved in clotting. I don't think I'm going to have time to go through the clotting process, but we'll give it a mention this morning when we're looking at defense really against infection. And then we started looking at transport of respiratory gases, and we got through the oxygen part okay, but the carbon dioxide part I need to add to the end of this. So the transport of oxygen involves hemoglobin. Structurally, hemoglobin's made of 4 subunits. A single unit in hemoglobin has got the 2 parts to it. It's got the protein globin chain, and the heme group inside that that's got iron in it, and it's the iron that can carry oxygen. So a whole hemoglobin molecule with its 4 iron atoms in it can carry 4 molecules of oxygen. Consequently, we have this little equation thing at the bottom here, where a single molecule of hemoglobin combines, or associates is the word that we would use, with 4 oxygen molecules in the lungs, and that association word is quite useful. We can represent oxyhemoglobin as HbO8 because of the 8 atoms in there, but you will see it written HbO2 or just oxy-HB. And then, of course, in the tissues that are respiring and needing that oxygen to be released, the oxyhemoglobin can dissociate in the tissues and release this oxygen back to release the hemoglobin to go and pick up some more. And the influences on that were where we then met the idea of partial pressure being important. So if you've got a mixture of gases, and that could be in a gaseous state, or it could actually be in solution, then the contribution of each gas to the total is related to the percentage that's there. So we measure oxygen concentrations, if you like, as partial pressure of oxygen, or oxygen tension is a term that's often used. This rather gloriously coloured version of the graph on your sheet, unfortunately the yellow, of course, didn't show up as well as it might have done, but that yellow shift is not an important bit particularly. The black line running through the middle is the standard sort of, well, you can either describe it as a hemoglobin oxygen association curve or an oxyhemoglobin dissociation curve. Remember, it works both ways because of that little equation we had on the last slide. Both things are happening here. We describe hemoglobin as being fully loaded at 95% because you'll always have some hemoglobin unloading its oxygen. So the oxygen tension, or the partial pressure of oxygen, where that happens, is this green line, and where it hits the bottom there is the loading tension. So we need that much oxygen. We need that partial pressure of oxygen to get the hemoglobin fully loaded. The blue line there at 50% is our sort of turning point, if you like. To the right of that, oxygen tension's above that, we can assume that the hemoglobin is more likely to be loading up with oxygen. But the oxygen tension's below that, we've got the oxyhemoglobin more likely to be giving up its oxygen. The red line and the yellow line are indications of how you can modify the behaviour of the hemoglobin depending on the biochemical conditions in the body. The yellow one, less significant, but the red one, the shift to the right there, described as the Bohr shift, is what happens if you get a rise in the partial pressure of carbon dioxide, which results in a drop in pH, of course, or a rise in temperature. And all those things, of course, are associated with exercise and muscle contraction. And what happens to the curve is the shift to the right makes the hemoglobin more likely to release its oxygen. I should say the oxyhemoglobin more likely to break down. We describe it as hemoglobin having a lower affinity for oxygen. And the unloading tension here, this vertical blue line, has shifted to the right, showing that the release of oxygen into the respiring tissues is actually made easier, I suppose, is the best description, by the effects of exercise. So it's a very useful effect, that the biochemical changes in the body make it more possible for the oxyhemoglobin to dissociate and to supply the oxygen that's needed. The shift the other way is just the reverse, but that was a less relevant example. And the other things that we met, you've got two other variations on a theme with the graph. We had the red line there represents myoglobin, and the myoglobin structure is like a single unit of hemoglobin. So hemoglobin's got these 4 subunits. Myoglobin is just one of those, and you find it in muscle cells, and that's the only place it is, and that's what makes red meat red, and it holds oxygen for the contraction of muscles, so it's supply the oxygen for aerobic respiration. Less significant, that bit, that doesn't come up in the exam, but understanding how the blood hemoglobin can change in behaviour is quite important. So also included the situation with fetal hemoglobin, because the mother and the fetus, of course, are sharing an area of gas exchange in the placenta where you've got one partial pressure of oxygen. Now, if that partial pressure of oxygen determines, let's say, that the blood unloads, well, the mother's blood would unload, fine, but the fetus would also unload. Well, that's no good. You need the fetus to have hemoglobin that will load with oxygen when the mother is unloading. So this graph shows you that fetal hemoglobin is different. It has a curve that is somewhat to the left of the mother's hemoglobin, which means that it loads at lower partial pressures. And there's quite a significant difference. So where the mother's hemoglobin is unloading, the fetal hemoglobin is loading with oxygen. And after a baby's born, it gets rid of that and replaces it with the adult version. But, it's obviously an important adaptation before birth. So that's covered the oxygen transport, and that leaves us with a little bit to do then on carbon dioxide. And you've got a fairly hideous looking sheet on carbon dioxide transport. Which is, the title for it at the bottom, the transport of carbon dioxide from tissue to lung. What you've got in the middle there looks seriously scary. I'm not going to use all of it. But you've got numbered boxes that tell you what's happening at any given place. And I think I'll go through the first part of it just in terms of getting carbon dioxide into the plasma. And then you can just assume that the reverse process will happen when the blood gets to the lungs and we need to breathe it out. Okay. All right. So yeah, it looks like this. All right. So it's after the hemoglobin ones. That's the one. Yep. All right. Now over on the left-hand side there, you can see a mitochondrion. Ah, for something you recognise. And the mitochondrion on there, you can see that effectively it's taking in oxygen and giving out carbon dioxide. All right, so we need to supply that oxygen. We've talked a bit about that, but we need to get rid of the carbon dioxide, because the problem is carbon dioxide, if you allow it to get into the tissue fluids, it will make them more acidic, and that will have an impact on enzyme activity, and so we have to get rid of it. So, right, let's see. I'll draw a sort of similar version of this, but just that first part. So if I start with the mitochondrion over here, that simple one, because we're not fussed about those at the moment. So that's inside a cell. And then we have a blood cell over here. It's got to be quite a big one because I've got to write some stuff in it. I'm not sure I've left myself enough space now. Let's move that up a little bit. So the important thing is then that if the mitochondrion is producing carbon dioxide, we've got to transport it to the lungs. So it's got to get into the blood. Now, I've already said carbon dioxide travels in the plasma. It doesn't travel in red blood cells. So why have we got a red blood cell? You'll see. There's some processing that goes on in there, basically. So the carbon dioxide that's produced in the mitochondrion diffuses out, and it ends up in the plasma. All right, so it goes there. I'm just trying to follow the diagram at the moment. It then has three alternative fates. You'll see that a very small amount of it travels in solution in the plasma. All right, so 5% of it you've got travelling in the plasma. That goes up to the top there. I'm not going to draw that on here because I'm not bothered about that bit. 10% is actually carried inside the red blood cell. All right, so although I said, that's it's not significant, there is 10% of the carbon dioxide produced goes in the red blood cell. But the other 85% is what I'm going to concentrate on here. Now, that 10%, let's go back to the 5% then in solution is literally just dissolved carbon dioxide. The 10% that travels in the red blood cell attaches itself to hemoglobin, and it produces a compound with one of my favourite words as a name. All right, if you look at it there, you've got HB, NH, COOH. All right, the NH is an amino group, and the name of that compound is carbaminohemoglobin. All right, so it's one of my favourite words, carbaminohemoglobin. And so that is just a way of transporting 10% of the carbon dioxide. The other 85%, however, we've got to process significantly. And although it's going to end up in the plasma, it starts by being processed in the red blood cell. So if you look at it, you can see that it's gone in and it's reacted with water. Now, I was in two minds whether to do this, but because I mentioned this last week when we were doing respiration and talking about control of respiratory activity being down to hydrogen ions that came from this reaction, I thought I'd better put it in, really. This is not carbon dioxide dissolving in water. This is it reacting with water. And the first step here produces an acid called carbonic acid. And the reason that this has to, well, the whole process involves red blood cells is because that process involves an enzyme. And the enzyme has to be sort of isolated somewhere, and it's in red blood cells. And it's a very important enzyme, actually, and you'll see it named up at the top, carbonic anhydrase. And I'm going to attach that to this somehow. Let's put it in a different colour. So the enzyme that's involved here, carbonic, in relation to the carbonic acid over here on the right hand side, anhydrase actually means removal of water. An means not, hydrase is an enzyme involved with water. So that enzyme is actually named by the reverse reaction. Carbonic anhydrase. So what we're being told is this is an enzyme that removes water from carbonic acid, but it's also responsible for controlling the left to right reaction as well. Right, so it's a nice opportunity to introduce the idea of reversible reactions and enzymes as well. All right, so the enzyme involved here is inside the red blood cell. From there, you can see that the carbonic acid, the H2CO3, breaks up. It dissociates. Okay, we've got the two-way arrow again, and it breaks up into hydrogen ions and hydrogen carbonate ions. All right, so this is where the trouble's going to start. Now, the weird thing about the hydrogen ions, at this point, they are actually responsible for helping to release oxygen from oxyhemoglobin. If you look down at the bottom, you have, I have to fill it in down here, HbO2, they've written oxyhemoglobin as. And we've got a two-way arrow with that. Oh, it's really hard to actually draw it right to left. That process to release the oxygen that's required is actually encouraged by the presence of these hydrogen ions, because they will combine with the hemoglobin. Those two come together to make something that is rather bizarrely described on the sheet there as hemoglobinic acid. You know, that's not even a real word, is it, really? But it is. It just means hemoglobin with a hydrogen attached, because of course hydrogen is the defining component of an acid. So that hemoglobin is released from the oxyhemoglobin by the presence of those hydrogen ions, and of course that frees up the oxygen which can go to our mitochondria. However, we're not really wanting to deal with that. What we need to do is to go back to that hydrogen carbonate ion over there that I've left dangling, because that's the carbon dioxide that we need to be transporting, and that's going to end up in the plasma. Now, as these hydrogen ions get used up, the hydrogen carbonate ion actually does diffuse out into the plasma. I haven't really got room for it here. Let's put it down there. The problem that we now have is actually that we've got an electrochemical gradient. By losing hydrogen carbonate ions, which are negative, we could end up with positively charged red blood cells. Now, that's not really going to work, because if all our red blood cells were positively charged, they'd all repel each other, wouldn't they? And that wouldn't really contribute to a very effective circulation if all your red blood cells were trying to get away from each other. So the diffusion out of these hydrogen carbonate ions is actually balanced by another negative ion diffusing in. And you'll see it down, you won't see it down anyway, you'll see it in the middle between those two red blood cells. The hydrogen carbonate ions are replaced by, can you see them? Chloride, that's right. Chloride ions diffuse in, and it's just, you can see it labelled on there as the chloride shift. You don't need this for the exam, but it's all right. It's just the chloride shift is the chloride ions diffusing in to take the place of the hydrogen carbonate ions that have diffused out. So there's a bit of an exchange going on there. Those chloride ions were originally hanging around in the plasma with some sodium ions. Sodium and chloride, very often associated with each other. So it leaves the sodium ions behind, but they're perfectly happy to hang around with the hydrogen carbonate ions. All right, I'm not going to put those on here. They're not included. Well, I don't know, because they're not included in the diagram sheet. Doesn't mean that they're not there, but the you'd have Na plus Cl minus. Rather than just writing NaCl, it's sometimes a bit more helpful to actually write the two ions. So those would end up there, and then these will hang around together then. So you effectively get NaHCO3, but again, they'd be ions. Okay, so you get sodium hydrogen carbonate is the sort of balanced version of it. So most of the carbon dioxide, this is our 85%, all right, here. All right, so this is 85% of the carbon dioxide that's produced by mitochondria in respiring cells, travels as hydrogen carbonate. I didn't need to draw all that out. Sometimes all I do in this section is just literally write 5% in solution in the plasma, 10% attached to hemoglobin, 85% as hydrogen carbonate ions in the plasma, and that's it. All right, but you've got the diagram there, and it's sometimes just helpful to demystify what appears to be a ridiculously complex sheet. Of course, if you follow what's on that sheet, then of course I've only covered half of it. On the right hand side, you've got another red blood cell, and of course it's just looking at the reverse process. So when the blood gets to the lungs, of course all that goes into reverse. So chloride ions will come out of the red blood cell. That will make hydrogen carbonate ions go in. They'll combine with hydrogen ions to form the carbonic acid. The carbonic acid will break down. All right, we will have this arrow when we get to the lungs. All right, the right to left bit. Carbonic anhydrase will break the carbonic acid down to carbon dioxide and water. The carbon dioxide will then be released back into the plasma. It won't go into mitochondria, of course. It does stop at that point, but it will diffuse out of the plasma, as we had on our diagram from last week with the little alveolus with the blood vessel around the outside that we had the carbon dioxide in the plasma. Once it's there, it can just diffuse out into the alveoli and be breathed out. Yay, you know. And we just thought the blood carried oxygen and carbon dioxide. Yeah, well, it's, you know, life is never quite as simple as it seems, is it? So that finishes off the transport bit. So we can move on. Now, the next part, I think, is, yeah, it's largely PowerPoint. So I will briefly stop the recording and load up the PowerPoint. Okay, so you've got a copy of these slides in your handout folders then, and there are some which have space for drawing little bits on, I think. Not very many, but we'll fill in one or two bits as we go. And of course you can annotate anything that seems useful. And so this will be a, you know, a fairly sort of structured trip through the immune response. Okay, first of all then, we need to be aware of the word antigen. Antigens and antibodies, completely different things. Antigens, well, the word comes from antibody generation. And an antigen stimulates the production of antibodies. Now, antigens can be a wide variety of different types of substance. We'll come on to that on the next cell. This is literally just a definition. And an antigen literally just stimulates the immune response. So everything could be identified by antigens on the surface. You know, individual cells are identified particularly by molecules that are attached to the cell membrane, but non-living materials have surfaces as well. Anything that is on a surface can act as an antigen. And then, of course, there are certain antigens that we refer to as allergens that produce allergic responses. And an allergic response is a form of immune response. All right. Very often it's an inappropriate one, isn't it? And it, you know, it can be, you know, a real problem to some people. But the immune response itself, of course, is a really useful thing. So this idea then, antibody generation, okay, the anti bit there and the gen bit there gives us the word antigen. And in terms of the types of substances, that's a huge list of things there. So we've got three different categories. Usually then, if we're looking particularly at living cells, no, actually saying living cells is not appropriate. Cells, okay. Now the reason I've changed that, you know, taken the living out of my sentence, is because how do vaccinations work? We actually inject bits of dead organisms, don't we? So just cells, all right. So if we're looking at cell surfaces, then what do we find? You know, we've got the membrane there. We might have cell walls as well, of course. You know, it's only animal cells that don't have a wall around the outside of their membrane. But they'd be the sort of things that we would find attached to membranes and walls. So proteins, not unusual at all. Proteins are built from amino acids, and they can be very small or very large. And we met them briefly when we were talking about hormones. Or polysaccharides. Now, what's a polysaccharide? Well, poly means many, yeah, multiple. And saccharides is actually sugars. And a polysaccharide is chains of sugars. All right, so something like starch would be a polysaccharide. Not that starch in itself is usually going to be an antigen. But we have chains of sugars on the outside of red blood cells, for example. That'll be one that we'll use when we talk about blood groups. The sequences of sugars on the outside of red blood cells determine whether it's A, B, AB, or O. So it could be parts of bacteria and viruses and other microorganisms. So it could be coats, for example, around viruses and bacteria. Capsules you get in bacteria, cell walls in bacteria, flagella, which are the little propulsion sort of strands that you get in some bacteria, And typically toxins, bacteria, tend to make us ill by producing toxins. Viruses get into our cells and disrupt the processes in the cells, but the bacteria produce toxins, either while they're still alive, which, is, I'm trying to think, those are exotoxins, because they're secreted from the bacteria, or as they start dying off, in which case they're endotoxins that come from the inside of the bacterium. But those toxins are then the antigen. The bacterium itself probably, would have carried some antigens, but didn't make us ill, but the toxins do. So they can be parts of microorganisms. We get antigenic properties in other molecules. Lipids, for example, well, you couldn't really have lipids as antigens because our cell membranes, of course, are all phospholipids, aren't they? So a lipid isn't going to be an antigen in its own right, usually. Similarly, with nucleic acids, are not usually antigens on their own. But if they attach themselves to molecules like proteins or polysaccharides, then they can be. So we can have some very elaborate molecules involved here. And then we've got these, this third category here, non-microbial, in other words, not from microorganisms. Exogenous. Well, exo means outside, yeah. So generated outside, and what we describe as non-self antigens. We have self antigens and non-self antigens. Obviously, the self ones, we need to know they're ours, so we don't attack them. And most of the time, we're quite good at not attacking our own antigens. However, there are certain diseases where we do. Yeah. 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 What we call autoimmune diseases. Yeah, variety of those. Type 1 diabetes. The body attacks its own antigens, but just in a particular area. Just in the pancreas, just in the beta cells that produce insulin. So yeah, we get it right most of the time, but then yeah, we have got certain diseases where we do attack our self antigens. Certain non-self antigens, of course, can cause responses, and so the list we've got there: pollen, egg white, we think of as causing allergies, yeah, allergic reactions. So hay fever with pollen, egg white being a well, I haven't put it in there just because of its sort of food link, really, because there are lots of allergens in food, aren't there? It's one of the things that my husband tears what's left of his hair out about, that actually trying to ensure that food production that says it's free of this allergen and that allergen actually is, and if it's not, then it has to be labelled properly, doesn't it? But egg white is also a significant allergen in vaccinations. If you have something like flu vaccination, they will ask you if you're allergic to egg white or eggs generally. Reason being, they grow vaccines in eggs. They grow vaccine in hens' eggs. Because if you're using a modified virus, usually it would be that sort, has to be grown in living cells, so they use eggs for that. So if you're allergic to eggs, then it can be a problem, but it shouldn't be, because the vaccine itself shouldn't actually contain egg protein. But there you go. Proteins from transplanted tissues and organs are on the surface of transfused blood cells. So here we are talking about human antigens and what we call tissue typing. And there's a whole set of genes that are responsible for tissue types, and they are what identify our cells as ours. And you could start off with blood grouping. That if you needed a kidney transplant and your group is B negative, then you'll have to have a B negative kidney for a start. But you can then tissue type a lot further than that, and you can get closer and closer and closer matches. So that's the antigen side of it. They are the trigger for the response. All right, they are quite specific. They are what cause an actual reaction. Okay, now the next bit is about antibodies, and I have a feeling that the diagram on your sheet's okay. All right. It is, actually. I don't know what version Oh yeah, that is the one. No, it's the next one that's actually come out a bit pale. Yeah, I'm looking at the wrong one there. This is a fairly detailed diagram of what an antibody looks like. There will be as many antibodies as there are antigens. All right, because for every antigen, you can produce a specific antibody. All right, now it's not just any sort of specificity here. It is actually stereospecificity. Yes, it is to do with shape. The shape of an antibody is determined by the shape of the antigen. All right. That'll be a bit more obvious on the next slide, although we will have to modify the diagram a bit, I think. You will never have to draw a diagram like this, or, well, not on this course. Obviously, if you know what you go on to, you are going to see, some of you are going to meet much more elaborate diagrams than we use. But all antibodies have this basic structure. They are Y-shaped, okay? And they have these double protein strands that just form this, sort of trunk with two arms sticking up off it. And the important bits are here and here. These are the bits that are stereospecific. These are the bits that are going to bind to the antigen. So it's only those bits that vary. So they're labelled as the variable region. Now, the actual chains here, heavy chain, light chain, doesn't matter. And usually you'll find antibodies drawn in exam questions just as a little Y, like that. That's all there is to it. All right, you might get a little bit more detail, which is really what we get on the next page, but you know that is enough. Now, if we look at the one that's come out a bit pale on your sheets, all right, you can sort of fill in the detail from that one. You can see it's the same diagram, but obviously we've lost all the labelling off there. But the important thing is this is just showing you that binding area, that variable area on the arms of the antibody, is shaped to fit certain antigens, but not others. And I love this diagram because I just think that looks like a row of 6 pairs of pants at the top there. Which ones today? Too much information. So this one here, the yellow one, is going to fit in there, but of course the others won't, because the binding site on the antibody is not the right shape. So we've got the rounded shape on this one fits into the rounded shape on the antibody, but the one above it, the purple one here, has got a stepped shape, so it won't fit in. The green one has got a sort of triangular pointed sort of shape, that won't fit in. So an antibody is produced in response to an antigen. We have to have cells that actually take in the antigen bearing material and produce antibodies in response, and that's really what some of the lymphocytes are up to. So even that diagram is more information than we need, but it's this specificity, this stereospecificity that is really important. We don't just have, hang on, specificity. The stereospecificity of the shape is, it's critical. We don't just have antibodies as a general thing. It's not like having, I don't know, a plaster you can stick on any part of your body to stop bleeding. Antibodies have to be specific. So that would be like having, I don't know, different shaped plasters for different bits of the body. And now I'm thinking about that, you do get different shaped ones, long, thin ones, square ones, round ones. Bad example. Never mind. You get the general idea. That you've got to produce these once the antigens got into the body. They won't just be there. Okay, it's the presence of the antigen that is going to cause the production of a stereospecific antibody. All right, that's the key to this. Some people are better at it than others. Some people fight infections much more quickly than others, don't they? And it is, the size of the response can vary between people. And particularly if you've already had an infection and you're left with memory cells. Some people's memory cells are more effective than others as well. Okay, so that's how our antibodies work. I realise I've got a blank slide there, and I've absolutely no idea what goes on that. It might just have been there just to show that you only need a simple diagram, perhaps, that you could just literally draw your antibody as a. I've got to have something to go on here, haven't I? We could have a little square space on there, but it will be the same. Whoops, that's a terrible diagram. But there we go. That's all I'm going to draw on there. And you don't need to draw it as two strands. So here are the antigens that have triggered the production of that antibody. There are some others lurking about, all right, but these are, not going to fit in there. But these little square rectangular, that's an appalling diagram, they will fit in there. Now, of course, they're not floating around loose on their own, are they? These antigens are on the surface of something. So, for example, a bacterium. Oh, I could use the space for a diagram like that. Let's say, here we are, we've got a bacterium. In fact, we might have several of them. Let's have 3 bacteria here. They're all going to be the same one, although I've drawn them different shapes and sizes. And let's say they've got little triangular antigens on their surface. Oops, I didn't need to draw that many perhaps, but yep, I can't draw triangles all of a sudden. All right, so if these are our bacteria with their antigens, then the antibodies, which are produced in response to the presence of these antigens, will, sorry, do them a bit quicker. What the antigens have done then is to trigger the production of antibodies that will attach to those. And they will come in, and I'm going to have to draw them sort of fairly inaccurately, but let's say that we have one that comes in here and attaches to those 2. Let's say we've got one that comes in here and attaches to those 2, and another one here that attaches to those 2, and another one here that attaches to those 2. And so what the antibody has done, or the antibodies, right, so these represent antibodies. Whoops. In this particular example, they have clumped the bacteria together. So the bacteria can't now infect cells. They can't now function normally. And they've actually been clumped into a form that makes it a lot easier for cells like the neutrophils to come along and destroy them. It's not just neutrophils, actually. There's a bunch of lymphocytes called killer cells, which are involved in this as well. But the antibodies are very good at immobilizing bacteria, or they could be neutralizing a toxin. If these bacteria, for example, had been producing a toxin that was little green circles. All right, let's say there's a whole load of little green circles have been produced. Then you might also have antibodies which come along. This time I'll draw blue ones. All right, so they might come along and immobilize, or not immobilize toxins, but neutralize toxins. Oops, I shouldn't draw it as a. So, okay, so the green bits there. Should make it clear what they are, shouldn't I? Those are toxins. That's the toxin. And that's a bacterium. And that, therefore, would be an antigen. Oh, that's made good use of the space. Not a particularly good diagram, but hopefully you can see now what the antigens are doing in terms of triggering appropriate antibodies, so the production of appropriate antibodies, matching stereospecific antibodies, and those antibodies then work by immobilizing microorganisms, neutralizing toxins, that sort of activity. Now, The next slide that you've got is a molecular model of an antibody, which doesn't really tell you very much about the different parts of it, but it's just, you know, showing you that the way that the protein molecule has been modelled gives us this this area down at the bottom with the yellowy green bit, and then the red bit at the bottom is the sort of common area to them, and then going up into the green parts of the branches, and the blue bits are the variable area. And yeah, I mean, it's not, you know, particularly useful in itself, but molecular models are often quite pretty looking things. And I think probably what you do get from this is an idea of the size. These are huge molecules because they're proteins, and proteins are enormous. All right, so antibodies are big. Now, moving on then to the actual immune response, we've got two parts to this. Triggered by antigens, all right, that's the whole point, generation of antibodies, so that the antigens are the trigger for it. And then we have these two parts to it. Now, they do get very complex in detail, but we're just going to sort of do an overview, really, of what goes on here. The first part of it we describe as the cellular response. And so it's about the production of cells. And the cells are known as T cells. To be specific, they're T lymphocytes. The T stands for thymus. And the thymus gland is in the chest. I can't remember whether, I think it's just in front of the breastbone, the sternum. And they're called T cells because that's where they mature. That's the purpose of the thymus gland. The thymus gland is particularly active in children when they're building up their immunity for the first time. After puberty, it tends to sort of shrivel and shrink. And so as adults, we tend to have very little thymus activity, a little bit left, but we're not meeting very many new infections, are we, as adults? So we don't need quite so much activity there. So that T is for thymus. All right, it's the T there. Of course, you know, originally the lymphocytes are going to have come from bone marrow, because we know that. The particularly significant T cells are the killer T cells, properly known as cytotoxic cells, but we still do call them killer T cells, because that's what they do. You know, they hunt and destroy. They attack and destroy any cells that are bearing antigens that have been recognised as non-self. And so they are doing the same sort of job as the neutrophils. You know, they can actually engulf and destroy these invading cells or invading antigens, whatever they're on, to be honest. So the T cells, those are the most significant ones. There are others, all right? We will come across some others. The other part of the response we describe as humoral. We met the word humour, didn't we, in the eye as being the substance, if you like, in the front and the back of the eye. So humoral sort of means it's sort of within fluids, actually, I think would be the best description. These, this produces lymphocytes as well, but this time they're B lymphocytes, and the B stands for bone, because they actually do mature in the bone marrow. All right, so we've got T lymphocytes and B lymphocytes, and they do slightly different jobs. I mean, the T lymphocytes, the key thing there is the production of these killer cells, the cytotoxic cells. The B cells, they are mostly responsible for production of antibodies. The way that they do this is that they divide to produce what's described as a clone. Now, a clone is many copies of something. And the cells that they produce are called plasma cells. So once we've triggered the production of B lymphocytes, they then produce this clone, in other words, massive numbers of identical cells called plasma cells. And it's the plasma cells that make the antigen-specific antibodies. So the original stimulus, sort of, it primes the cells to produce the right sort of plasma cells. The plasma cells then churn out the antibodies that are specific to the antigen. So you get, you might only need to sort of produce one lymphocyte. That might then produce a clone of 100 plasma cells, and then they churn out millions of molecules of antibody. So the antibodies, we know now that they help to destroy antigen-bearing cells. Not necessarily actually destroying them, because the antigen is only a protein after all, and for a protein to actually destroy something, not really a mechanism for that perhaps, but it does, they do clump cells together, or they can actually damage membranes and that sort of thing to help with the process. Now, the important bit as far as retaining immunity is concerned is the production of memory cells. Memory cells are produced, well, you get T cells and B cells produced as memory cells, and they retain a memory of, not a mental nervous system type memory of, but they are called memory cells, and they retain recognition, if you like, of the antigens that you were first exposed to. And they are the key to retaining immunity. So if you had, let's say, chickenpox as a child, and you recovered from it, when you were recovering from it, you produced memory cells. So you don't get it again, do you? Some people do. Chickenpox is I chose that because it is it's an odd example. I've heard of people getting chickenpox 3 or 4 times. And, of course, you can also get shingles as an adult. If the chickenpox virus goes dormant in the body, you can get shingles. And, so the memory cells are clearly not there or not working properly. So, memory cells are really key. And it's memory cells, the production of memory cells is the whole point of immunization. If you vaccinate someone, you don't want to make them ill. You don't give them the disease. You want them to make memory cells so they think they were ill, or their immune system. It can't think, can it? I've got to stop this. But the immune system has a functioning chemical memory, shall we say, of what appeared to be an illness, but it wasn't, because what the immune system remembers is the antigens. So as long as you put the antigens into someone, they don't get ill, but they make the memory cells, and so if they then encounter the chicken, if you can actually vaccinate against chickenpox now, can't you? Although it's not routinely done, but if you gave somebody a chickenpox vaccine, it wouldn't give them chickenpox, but they'd make the memory cells, hopefully, and then if they encountered it, the body would respond so quickly, so rapidly, and it produces a very large response that the illness wouldn't actually take effect. But I mean, you get this with everything, even colds. All right, you never get the same cold twice. There's about 200 different colds. So, you can start ticking them off, when you get to my age, there's not so many left to have. But, it's the reason that small children always have a runny nose. They'll pick up anything. And then you gradually start, recognizing the colds and building up memory cells. We don't necessarily remember them all, perhaps. But, we might think we're going to be ill. If you've been around people with a cold, you might get a bit of a sore throat, or your nose, you might wake up, bunged up nose. But then after a couple of hours, it's gone. And that's because, yes, something got in, but your body recognized it, kicked off really fast, the memory cells got in there, multiplied very rapidly, produced huge quantities of antibody, cleared it out, and you're okay. But you sometimes just get that hint of an infection, don't you? And then it doesn't turn into anything. So it's one you've had before. So this second exposure, it's the first exposure produces the memory cells, and the second exposure then causes a really rapid response and a very big response as well, so that you don't actually get the infection causing the illness. So that's the really sort of important part in terms of retaining immunity. Now, this diagram is not a good one. I really ought to replace it, and I think that to myself every year when I get to this point. I think, oh, that one again. But if you follow it through, you can see pretty much what's happening. There's one or two spelling errors on it. So we start off primary exposure to the antigen at the top. The antigen we can assume is, let's say it's on the surface of a bacterium or something, and a macrophage, well, let's say a neutrophil, a white blood cell, will engulf the bacterium, and the antigens will then actually appear on the surface. Actually, I'm not sure if it is the, no, it won't be the neutrophils, it'll be one of the others. The antigens appear on the surface, and we should, of course, only have one R in there. Yeah, antigen presentation. That triggers the production of the first type of cell that we get is in the T cell category, and they're called helper T cells. And, you know, it's funny, isn't it, how you can have all this really elaborate terminology, and then you get a few words like killer cells, helper cells, and you think, surely there must be a more complicated word than that. But the helper cells are, they do literally help the rest of the response to kick off. And it actually, they are helping both, I need a colour that's not on here. They are helping the production of B cells and T cells. All right, so these helper T cells are promoting the production of the B cells. Although, you know, we are getting to that point anyway, but the helper cells actually increase the response. So we get the B cells produce the cytotoxic T cells. So let's go over to the T cells because we started with those. They produce active cytotoxic cells. Those are the ones that are really going to go around clearing up. But of course, they will also produce memory cells. And if you get a second exposure to the antigen, then we get memory helper cells that will produce memory cells. And so this is a very rapid little response down at the bottom here, that, you know, with the second exposure. Over the other side, we get our B cells produce, now this, plasmacytes. I called them plasma cells, which of course is exactly the same word. Cytes means cells, so plasmacytes, plasma cells, same thing. And this is where you get the clone, the large number of these cells produced, which are specific to that original antigen. We also get, of course, the production of memory cells here, so that again, if we're exposed a second time, we get this really rapid production of plasma cells again. And it's the plasma cells that produce the antibodies. All right, so those antibodies are going to be very important to us. And the secondary antibody response down there at the bottom is what would happen the second time you're exposed to the same antigen. Okay, right, so that's a real sort of overview of it. I don't think I've put anything else in there. Now I'm going to stop at that point, so I have a slightly sore throat. I just need a cup of coffee, to be honest. So I'm going to stop at that point, and then we will okay, now in order to do this then, oh, it's going to do that, isn't it? So I can close it anyway. All right, I'll do it as it comes. I'll go back to, yes. Yep. Okay, I can put it on here then. Types of immunity. We can categorise in various ways. It's there are two ways and then two sub-ways of doing it, and you can do it in whichever way round you like, actually. I'm going to start by dividing immunity up into natural and artificial. Okay. Now, the natural immunity really is what we've just looked at. So a natural process involves the body responding to an infection, perhaps. Artificial would be getting the body to respond to something that's not actually an infection. So within each of those, we've now got two other categories. And on both sides, it will be an active form of immunity where the body is actually doing something, or a passive form where the body is just given something to make it immune. And on the natural side, the active form of natural immunity is recovery from infection. That's what we've just looked at, you know, the recognition of the antigens on the surface of organisms such as bacteria and viruses, the production of appropriate cells, and then the antibodies probably. So this would be recovery from infection. And so that's a natural process, and that's how we build up quite a substantial amount of our immunity. On the other hand, there is passive immunity where we don't actually have to do anything, where we're actually given the antibodies, and that's before birth. That babies are actually born with the antibodies that their mothers have. So at birth, babies are actually immune to the same things their mothers are, but that doesn't last very long, because obviously, you know, these are not going to stay in the system, because they've not got enough memory cells and this sort of thing. So this is passed from mother to baby before birth. And there's an obvious benefit there, you know, that you want to protect newborn babies. So it's quite a tricky thing. I mean, antibodies are quite big proteins, so for them to be able to cross the placenta, it's not easy. And so it tends to happen really late on, you know, just, I don't know what we're talking, sort of days or a couple of weeks perhaps before birth. So premature babies are at a much greater risk of infection because they haven't necessarily got all of that. The other transfer, of course, is through breastfeeding. The antibodies are, you know, part of that, bonus, really. The artificial side of it, we also have an active and a passive version. The active side is the majority of vaccinations, of course, trigger the body to produce memory cells. Yeah, and you can follow the history of it back to smallpox and Edward Jenner, who discovered that milkmaids who had picked up cowpox from milking cows didn't catch smallpox. And so he trialled a vaccination process where he took some of the blisters from a cow with cowpox, scratched a small boy's arm and actually introduced some of this stuff in. And the boy turned out to then be immune to smallpox. So that was the start of vaccination. Now, if tomorrow you choose to do, the essay choice that includes vaccination, that's where you start with the history of it. You know, that little story, is where everything started. And of course, since then, vaccination has come a huge way. In itself, it is, it's one of the biggest advances in medicine. I remember on a television program once somebody saying, what the three most important advances were. One of them was vaccination. The other one, I think, was anaesthetic. I can't remember what the third one was. Don't think it was antibiotics, because unfortunately we've messed up on that one, haven't we? It might have been. I think it might have been pain relief. But I think, the ability to actually kill pain is actually very important. So yeah, vaccination. Immunisation is a better term for it. Vaccination is actually a specific type of injection. The reason it's called vaccination is, of course, it comes from the word for cow, which is vacca. Yeah, vacca. Is that Latin, Italian, Portuguese? Yeah, I was going to say it has a. Yeah, I was going to say it has a. It has an origin in Latin-based languages. Vacca means cow, So yeah, a vaccination is is an injection of cow. But yeah, immunisation covers everything. But vaccination, I think, is specifically supposed to be where you introduce organisms, perhaps. I'm not entirely sure, a vaccine, but we use it much more broadly now. A vaccine is actually, it could be anything. And very often it's just antigens off the surface of organisms, or it's dead versions of the organisms, that they've been killed off, but of course they've still got their antigens on the surface, so they still trigger the immune response. So immunisation, I'm going to struggle to fit the other one in there, aren't I? Immunisation, where we've got antigens injected they stimulate the actual response. I was supposed to put the word active, wasn't I? Sorry. That's the active. Sorry about that. Oh no! Yeah, sorry, that was supposed to be active there. This is a very, very lopsided diagram, this one. But that stimulates the actual response, so that is active. Stimulates. Dear me. To stimulate response. So that's where we are getting an active immune response. You see, that's why you needed the word active in there. So yeah, sorry if you were one step ahead of me. And the other option, of course, is the passive one. Passive artificial. Now, that's an interesting one. Oops. All right, the eye has disappeared in there. Passive. All right, so what on earth could be a passive artificial one? It actually comes up on the exam, this one. And it's where you actually have an injection of antibodies. And there are certain situations where you can be given an injection of antibodies. Now, the antibodies, of course, are not always described necessarily as antibodies. They might, for example, be called an antitoxin, because antitoxins will be antibodies. They clear toxins out. So what situation might you need an injection of an antitoxin? Yeah, but particularly bitten by rabies, interestingly, you could have, but snake I was expecting. Yeah. Bitten by a rabies. Bitten by a snake. If you're bitten by a snake, the toxin, the poison gets into you, and you can be treated with anti-venom, antitoxin, and that is an injection of antibodies just to that particular venom that will help clear it out of the way. Because you don't get long-term immunity. You can be bitten by that snake again. You'll need another injection. All right? You don't build up an immunity from that. But other situations, and I'm not sure, but I know that tetanus is an example where you can be injected with the antibodies. If you, let's say you're in the garden and you put the fork through your foot, that's the classic, or you're on the beach, let's be a little more summary, and you stand on a piece of glass, all right, let's say you cut your foot badly, you go to casualty, and they say, are you up to date with your tetanus? And you say, I don't know, because very few people can remember. And you get it, if you don't know or you're not, you'll get an injection on the spot which has got anti-tetanus antibodies in it that will deal with any possible infection there and then immediately. But you would still need a vaccination to be up to date on your tetanus. That doesn't give you immunity, that's just the antibodies. And I think there are one or two others as well. I think there's a treatment if you're in contact with people who've got hepatitis B. I think you can be given, if you've come into contact with someone, I think they can do it for that. And the rabies is one, I'm not sure, because you can treat that quickly, I know. But the passive one, well, no, I know it can be treated quickly.
Is that what you do, because if say, for example, you set someone that had HIV.
The HIV one, I'm not sure. HIV, it might be, although the problem with HIV is that I think it's more of a suppressant. I think it's to suppress the activity of the virus, to be honest. I don't think it's antibodies. But it might possibly be. I don't know. It'd be, something you could find out. But the idea with the passive one is it's an injection of antibodies, but it doesn't give you immunity. So yeah, I shouldn't have really said immunity as the top bit of that. So it's injection of antibodies. All right, no memory cells. I'll put e.g. anti-venom. No memory cells made. So you don't have that long-term immunity in that case. It's an immediate protection, and that's it. It will clear out the damage at that point, but it won't actually protect you any longer. Oh, right, yeah. With hepatitis B injections, the problem with Hep B is it's very damaging to the liver. It's transmitted through body fluids, so it's on the same scale as HIV. So if you're going to work in healthcare, they do usually look for you to have hepatitis B in immunisation, and you would then be long-term protected against that. Yeah, it's quite likely, that you need to be protected wherever you could possibly come into contact with blood.